Redox reactions, detecting traces of blood. In a presumptive blood test, the Kastle-Meyer test, a sample is collected with a cotton swab. A drop of phenolphthalein is added, followed by a drop of hydrogen peroxide. If it immediately turns pink, the presumptive blood test is positive. During the process, the colorless phenolphthalein is oxidized by hydrogen peroxide at the presence of blood sample into oxidized phenolphthalein, which is a pink color. In order to understand the reaction mechanism, we need to first go over the redox reactions. A redox reaction is a reaction involves electron transfer. Here is a typical redox reaction. Zinc reacts with copper ion to form copper and zinc ion. In this reaction, zinc loses 2 electrons to form zinc ion. Zinc is oxidized. On the other hand, copper ion gains 2 electrons to form copper metal. Copper ion is reduced. Because copper ion is reduced by zinc, zinc is called a reducing reagent. On the other hand, zinc is oxidized by copper ion, and therefore copper ion is oxidizing reagent. In the Kastle-Meyer test, the phenolphthalein is oxidized by hydrogen peroxide, and the hydrogen peroxide is oxidizing reagent. The protein in red blood cells contains an organic molecule called porphyrin. A porphyrin molecule has 4 nitrogen atoms that coordinate with an iron atom, which is responsible for carrying oxygen in the blood. The combination of porphyrin and iron is called heme. Let's use PFE3 to represent heme with an ion that has an oxidation state 3+. When hydrogen peroxide is added, hydrogen peroxide is deprotonated to peroxide, which binds to the iron atom. Each oxygen is in the -1 oxidation state. Next, the oxygen-oxygen bond breaks, and one oxygen gains an electron and combines with protons to form water, while the other oxygen forms oxygen atom, which combines with heme to form an organic radical, where the molecule has an unpaired electron. In this radical, iron is oxidized to iron 4+. This iron 4 porphyrin radical is a powerful oxidant that can be able to oxidize an organic molecule such as phenolphthalein to its oxidized form, causing the color change. And at the same time, iron 4+ is reduced to iron 3+. The net change during the process is the hydrogen peroxide is reduced to water, and an organic dye is oxidized. What happens to the porphyrin iron complex? It starts with iron 3+, which combines with an peroxide and then forms iron 4+ radical, which goes back to its initial iron 3+. In other words, it participates in the reaction and makes the redox reaction possible, but overall it is not consumed. It serves as a catalyst and catalyzes the reaction. Only a very small amount of hemoglobin are required to give a positive result. In this experiment, you are going to perform presumptive blood test on several organic dyes. The first one is phenolphthalein with hydrogen peroxide and with the presence of blood sample. Phenolphthalein will change color from colorless to pink. The second organic dye is tetramethylbenzodine. This compound will be oxidized from colorless to blue. The third one you are going to test is leucomalachite green. This compound will be oxidized from colorless to green color by hydrogen peroxide in the presence of blood sample. The last one is luminol. The oxidized form of this compound shows chemiluminescence. Here is the list of chemicals you need for SDS. In this experiment, you are going to test 3 samples labeled A, B, C. One sample is treated with blood and therefore will test positively for each test. You will obtain 4 cotton squares for each sample and treat them with black light, phenolphthalein and hydrogen peroxide, leucomalachite green and hydrogen peroxide, 3355 tetramethylbenzodine and hydrogen peroxide, luminol and hydrogen peroxide. As mentioned before, you are going to apply 4 dyes with hydrogen peroxide to 3 samples. After the test, dispose all the treated samples into the solid waste container. Pre-lab questions. Question 1. It asks you, why should you not be surprised that plant enzymes can interfere with test for blood? We have shown in the mechanism slides, it is the porphyrin iron molecule or the CHEM that catalyzes the redox reaction. The blood samples contains CHEM and therefore it will give you positive test. But if a plant oxidates the protein that contains CHEM, it must also give you positive results. Question number 2 asks you to find MSDS for phenolphthalein and leucomalachite green. Find out their health concerns and precautions. This should be covered in your SDS section of your lab report.